Uh, Good morning, everyone. Thank you for joining this webinar this morning. My name is Azadeh, and I'm from Decode Science. We are local distributors of MGI Genomics in Australia. So with me, we have Dr. Sen Wang from the South Australian Genomics Centre, and also Dr. Haofei Wang, who is the field application scientist from MGI Australia. And together we're very happy to share the latest. Well, first of all, be able to introduce MGI as a sequencing technology, and also, I will get started with just giving you a brief overview of the technology, of the DNBSEQ technology, and how it's applied for sequencing. After me, Dr. Sen Wang will be telling you about their experiences at the South Australian Genomics Centre with the technology and what they've been using it for and what their experiences so far has been. And finally, we have Dr. Haofei Wang, who will be introducing CoolMPS, which is the latest advancement from MGI, and it's a very exciting new part of the technology that's just being introduced and being we are getting started with that technology in Australia, so we are very excited about that. So just before I get started, please please feel free to put your questions in the Q&A section, and I will keep an eye on the Q&A section, and after all the presentations are done, then we should have 10 minutes for Q&A. So I'll keep an eye on the chat box as well as the Q&A box, and I'll be asking your questions and making sure that your questions are answered. Okay, with that, I'm going to get started with giving you a very brief overview of MGI, who is MGI, and also what DNBSEQ technology is. So some of you may have heard of BGI. So BGI, or Beijing Genome Institute, are one of the biggest genomic research institutes in the world, and they have a number of subsidiary groups who are active in different areas such as agriculture research, genomics bank, and also they have a journal, Giga Science, and BGI is one of those subsidiaries, and what they do, they are the manufacturing arm of BGI. So they manufacture the genomic sequencing technology as well as reagents that are applied for, they are basically providers of genomic sequencing technology to BGI and also the rest of the world. So BGI acquired Complete Genomics, which is the basic of the MGI DNBSEQ technology some years ago, and they did some further development and did some more R&D work, and now it's there are multiple. The MGI R&D is headquartered in Shenzhen in China, but there are branches worldwide which are contributing to continued R&D and as well as providing application and also service to users worldwide, including Australia. So with that, a little bit of introduction. I'm just going to get into my introductory presentation on MGI. So we're going to talk about, I'm going to talk about how the library prep works, and also what the DNA nanoballs are and how the sequencing actually happens, and yes, and how it differs from other technologies that you may have used so far. So starting with the library prep. So I guess all of us are familiar with the steps involved in next generation sequencing library prep. So you, for example, if you're doing whole genome sequencing, your workflow starts with some sort of DNA fragmentation, followed by DNA repair adapter, A-tailing adapter ligation, and then with an optional, well, just depending on the workflow, with a PCR to amplify the final library. And then your final library would be a double-stranded DNA, piece of DNA, like something like this, with adapters at ends, and then sequence areas where you could hybridize your sequencing primer. So MGI library prep does involve all of those steps, but it does with an additional step of heat denaturing double-stranded DNA to have a single-stranded DNA, and then using a splint oligo, which is complementary to your our adapters at both ends of the library, to create a circularized library. So this is a single-stranded circularized DNA, and that's considered to be the final library molecule for your MGI sequencing. So this is a typical structure of a simple MGI library. You have your insert, which you're planning to sequence, and then you have your barcode here. You have a area here where your barcode primer would anneal and start sequencing the barcode, and you have an area here where your read-one primer would anneal. So this is very briefly introducing a typical MGI library. So now we go to DNA Nanoball Generation. So DNB stands for DNA Nanoball, and so what is DNA Nanoball and why we use that? So let's say you have your single-stranded circularized DNA, and if you try to sequence using just one single copy of that, you will have a rather weak signal, which you may not be able to capture it with the camera equipment on the instrument. However, if you amplify that, and that's what we do with Nanoball Generation, we create many copies of that DNA. Now we have an intensified signal, which is going to give us higher signal intensity and better quality data. So that's why we amplify the DNA, the library, to create DNA nanoballs, so that you can get a brighter signal that can be captured with camera. So how do we do that? So the process of DNA nanoball generation happens on a thermal cycler outside the instrument. So what we add is that we add a polymerase and some primer to our library, and then after the primer is annealed, then it is extended, and it's going to amplify the whole library. And then when it reaches the first, the same where it started, it's going to displace the original strand and keep copying your library using a rolling circle amplification. So what are the advantages of using this rolling circle amplification? The main advantage is that for every cycle of amplification, which we're going to do 300 to 500 cycles of amplification for each molecule. For each cycle of amplification, the template is the original DNA or library sequence. So we're not going to get any if there are any errors happening due to PCR amplification, that error is not going to get amplified over and over again. So that results in higher sequencing accuracy and low amplification bias. So after we generate the DNA nanoballs, then we're going to load them onto flow cells. So this is a typical MGI Seq flow cell. And what's on the flow cell are these patterns of spots where DNA nanoballs sit, and they are sequenced and imaged. So these spots have been treated with amination modification, so they are positively charged, and the pitch, which is the center-to-center distance between these two, every two spots are 750 nanometer, while the diameter of every spot is around 220 nanometer. And by the way, the area outside these spots are also treated chemically so that DNA nanoballs cannot bind to them. So when we start loading our DNA nanoballs, because our DNA nanoballs are negatively charged, they're going to bind to these spots using van der Waals forces. And as I mentioned, they cannot bind to areas outside these spots. And so the spot diameter, as I mentioned, is around 220 nanometers, and a typical DNB diameter is between 220 to 240, so could be slightly larger than a spot. So we cannot fit more than one DNA nanoball in every spot. After the DNA nanoball is are sitting securely on the spots, any extra DNA nanoballs are washed away, and we start the process of sequencing with combinatorial probe and core synthesis. So what it involves is that nucleotides or bases are incorporated into our sequencing process, into our DNA nanoballs, and then because they are fluorescently tagged, they can be imaged, and then those fluorescently tagged nucleotides are cleaved, and we go back to incorporating new bases. So at every cycle of the base at every cycle of the sequencing, we can detect one base which is going to be analyzed and base called by the instrument. And then the final output is a FASTQ file, which can be analyzed with any existing pipeline that you may have. So there isn't any special pipeline, analysis pipeline for any application from MGI equipment. And you can also do paired end sequencing. I realize I'm a bit over time, so I'm not going to go into very details, but the same. So we're going to the same way that we do a single end sequencing, we can just add an extra multiple displacement amplification to create strands for the second read, and then do the paired end sequencing. The advantage of doing this with the MGI platform is that you get equally or almost equally bright signals for the second read as well as the first read, so that increases your data accuracy and quality. So just very briefly introducing different sequencing platforms from MGI. So we have a few different platforms with different outputs for depending on the throughput that every center might need. So that starts with the desktop sequencing instrument G50, which produces 150 gigabases of throughput per every run. And there we have a medium throughput sequencing instrument G400, which produces just over 1 terabases of data in every run. And then we have a production scale sequencer T7, which produces up to 6 terabases of data in every run, and that's really suited for whole genome sequencing projects and deep exome sequencing projects and so on. We also have a latest sequencing platform, which is a TX. So that involves a very cutting edge technology of doing large scale sequencing using very large flow cells that actually use recycled sequencing reagents. And it's what you can see here is our multiple imaging instruments and a biochemical reaction center here, and there is a robot that moves the flow cell between the biochemical reaction center and the imagers. And using this platform, we can sequence 50,000 whole genomes every year. So this is for very large scale population genomics, and that significantly reduces sequencing cost. So we can look into doing a whole genome, a human whole genome at 30x coverage for a cost of 200 Australian dollars, which is really exciting. So this was my introduction. Thank you very much. I'm gonna stop sharing and let Dr. Sen Wang share his presentation. And while Sen is doing that, I'm going to just briefly introduce Sen. Dr. Sen Wang received his joint PhD degree from Shanghai Medical School and University of Basel, and has more than 10 years experience using NGS technologies with an in-depth understanding of its applications in the research and clinical fields. Sen was running the NATO-accredited genomics facility back at Monash Health Translational Precinct over the past 5 years, until he recently took on his current role at the South Australian Genomics Centre. Thank you, Sen, and I hand it to you to start.
Can everyone hear me?
Yes, I can hear you.
Yeah. Thanks, Azadeh, for the introduction, and it's a pleasure to be here today to talk about, to briefly introduce the newly established South Australian Genomics Centre, short for SAGC, and also our experience with the Mgi sequencing equipment. So the title of my topic is Partnering Genomics and Bioinformatics at the SAGC. So the SAGC is a statewide genomics facility which was recently established. And actually we opened our shop in July last year, 2020, as a part of a 7 million investment in genomics and bioinformatics. We are a not-for-profit genomics facility and also a unique partnership between SAMRI, University of Adelaide, Flinders University, UniSA, the Australian Wine Research Institute, and AGRF. And we are also funded by Bioplatforms Australian, which is enabled by NCRIS. I think our goal is crystal clear, that is to provide end-to-end and cost-effective, readily accessible genomic services to local research, clinical, and commercial communities. So as a genomic facility, we offer a list of services majorly divided into two parts. The first part is genomics. We offer from sample extraction, next generation sequencing, and also we focus on single cell and spatial transcriptomics. And on the bioinformatics part, we have multiple pipelines which can carry out DNA-seq, which includes whole genome sequencing, whole exome sequencing, RNA-seq, including mRNA, total mRNA, single cell, spatial, etc., and metagenomic sequencing, epigenomics, and reduced representation sequencing.
Sorry, Sen, I'm not seeing your slides, so can you please share them? Maybe it didn't.
Oh, I thought I'm already sharing my slides. Okay, just one sec. Sharing.
Okay, it's there now. Thank you.
Okay, yeah. So just one sec. Okay, so yeah, I will quickly go through this, like the first couple of slides. This is the first slide, and this is I just talk about SAGC, the unique partnership between 3 universities, SAMRI, AWI, AGF, and we are funded by Bioplatform Australian, which is enabled by NCRIS. And this is our goal. And this is a list of services I just talked about, basically for 2 parts, genomics and bioinformatics. I will come back to this later, so don't worry. And normally, this is our workflow. If you want to initiate a project with us, first you just get in touch. We have our website, sagenomics.com.au, or you can simply drop an email to our SAGC email. Of course, if you are based in South Australia, you are welcome to just pop into Samurai or Adelaide West Campus of Flinders University to have an in-person conversation so we can get the project started. And then we will discuss about the experiment design. Then if you are happy, we will finalize the experiment design and provide a quote. Once you are happy with the quote, you accept the quote, and we organize the sample submission, sample processing, sequencing, and we will carry out the data QC, transfer, quality report, and of course, we value a lot about your feedback. And this part of the process, we are currently in the process of applying for NAT accreditation 17025 to make sure we are offering the highest standard service possible. Okay, so a little bit more about Ngs Applications as a sequencing facility. So as mentioned, we offer all sort of, including the standard pipelines and also customized pipelines, including RNA-seq, like mRNA, total RNA. We've also got in-house protocol which can handle RNA as low as 500 picograms starting from, and also target RNA where there's enrichment before library prep, small RNA sequencing. As for the DNA sequencing part, as mentioned, we do a lot regularly about whole genome sequencing, whole exome, target sequencing, epigenome, and metagenomics. And since we are at the MGI webinar today, I will talk about in my next 5 to 10 minutes, I will talk about more about the Mgi Sequencer. We installed Mgi G400 in June this year. So we've got, I think, up to now, about 5 months experience, hands-on experience with this equipment. And I think overall, we can say the data quality is quite satisfactory. This is a figure I got from MGI. As you can see, this is a 400 cycle single rate. You can tell the Q30, the quality score is really high with an average of 91%, low error rate, etc. And so in order to do a proper validation and do an apples to apples comparison, we use third-party library preparation method for different kinds of libraries, and we sequence them both on Illumina instrument and on MGI G400 so that we can have an apples-to-apples comparison about the performance from the two equipment. And this is the data from in-house. As you can tell, the quality score is really high. Don't get confused by that because the y-axis is actually really high. So the quality is the Q30 is all above Q30. This is from 32 to 36. Here in this figure, we compared 5 RNA-seq samples using an RNA-seq protocol. The red lines you can see here is Illumina, and the green ones are on Mgi G400. As you can see, the quality score is fairly comparable. Here, this is this first cycle, first couple of cycles. So probably just only focus on the middle part, and this is towards the end of the rate. So don't take account into this part. So if you look at the middle part here, the quality is fairly comparable between two different equipment. And also we looked at the duplication rate. Again, the duplication, this is the raw rates, which means this is the rates without deduplicates using UMI, unique molecular identifier. As you can tell, the duplication counts rates here, the distribution, it's pretty much overlap with each other from two different equipment. But we do notice if we use UMI for deduplicates, like the MGI equipment shows a better performance. As you can see, this is the red bar is 5 RNA-seq samples on Illumina. Like the duplicate rates is varies somewhere between 10% to more than 40%. But with MGI, it sits fairly low at around 2 to 3%. We are yet to identify, or we are keen to hear a bit more why this is the reason. So probably Haofei can talk about more on this in her talk. But this is what we've observed in the different sequencing runs. And we didn't stop there because we did a full race alignment and gene identification, genes profiling to make sure we are looking at the right thing. We did a thorough validation on the instrument. So this is again the observed genes with CPM above 1. As you can tell, when we do the alignment and the genes profiling, two technologies are very similar. The performance is very compatible. This is sample 1 to 5. Again, these are the 5 RNA-seq samples. Just keep in mind. And these are the table with specific numbers. The top 5 are the observed genes, like pretty much identified 17,000 genes from MGI and Illumina. It's very similar. I wouldn't say there is a significant difference in terms of this. And but assuming if the alignment rate is similar, but with a low duplicate rates observed here, I guess from a facilities perspective, that means in each run we will be able to multiplex more samples on MGI instrument. So that's our take-home message. And as I only showed the RNA-seq samples here, but we've also tested out a lot more other sample types, including mRNA-seq, total RNA, ChIP-seq, the whole genome bisulfite sequencing, whole genome amplicon. And recently, we spent a lot of effort testing single-cell RNA-seq and Visium, which is a spatial transcriptomics library type on MGI. And because the performance on these libraries, which colored green, are very similar, just as shown here as the RNA-seq libraries, I won't go into detail on that, but I will talk about a bit more about single cell and spatial transcriptomic libraries because we do bump into a bit of issues. And I'd like to hear more both from MGI or other users to see what their comments are. So this is an example of the 10x library templates. As you can see, when we do the single cell analysis, when we run the pipeline, the first difference you notice is the data format is actually quite different from Illumina's FASTQ file, from the MGI FASTQ file. This is the Illumina FASTQ file here, and machine ID, run ID, and this is the FASTQ file you get from MGI instrument. As you can see, the position of the information is actually quite different and the format is quite different. So you do need a bioinformatics team to carry out the customized analysis. And again, the library structure is slightly different from the MGI equipment. You will only get 2 reads from each lane. One is read 1 and the other is read 2. And your index is located as a tail of read 2, which is at the end of read 2. And if you read, this is I7, no, this is I5, I believe, and this is I7. So we read 10 bases of each for demultiplexing here. So this is the library structure. And for the single cell library that we tested here, which I'm going to show you in the next 2 slides, we actually only have one single I7 index. So which means we only use the 8 base pair I7 index at this position. So we discard the I5 bit and the first and these 2 bases at here from I7. So this is a single DEL index, and this is a Q30, as you can tell. With single cell libraries, the structure is the single cell index and the cell unique molecular identifier is in the first 28 base pairs. So you can see from the MGI rates, the quality is actually quite good, up to 28. And then this is like a waterfall, but it doesn't matter because what really matters in rate 1 is the first 28 base pairs. And from 100 cycle, this is the rate 2, which contains the mRNA transcript, which is the mRNA profiling rate. And you can see the quality of rate 2 is actually quite good, up to 200 cycles. And again, as I mentioned, we are only using I7 here, so which is the last 8 cycles here. So the quality is, again, really good. This is I5, so we will discard this part for demultiplexing purposes. And so the matrix we use here is more than 3 base pair differences between our samples, and we have more around 95% of rates assigned, allowing 1 base pair mismatch, which is a common threshold used for demultiplexing, even for Illumina equipment. But that is single cell libraries. But we do observe bumped into a bit issue with the spatial transcriptomics libraries, which is Visium. And again, the first 28 here we multiplex 2 Visium samples in this line, so which means there are only 4 indexes present in this line. And again, the rate itself, the quality is actually quite good. This is rate 1, the first 28 base pairs, and this is rate 2. But the index rate here is actually the quality is really low, and we can't have a very good demultiplexing rate using the indexes here. I'm not sure. We had a bit of conversation with MGI, and the possible reasons could be we only have 4 indexes here, and the diversity of the indexes are not great. But with these 4 indexes on sequencing on Illumina instrument, the quality is actually quite good. So the conclusion here, at least from this run, is it seems MGI instrument is a bit more sensitive to index complexity, and probably Haofei can also comment on this or maybe explain a little bit whether this is the case which leads to a low quality of the index rates here. So again, because the data format, what you get from the instrument is actually quite different. I know on the instrument, you can do quite some standard demultiplexing using the algorithm on the instrument, but as a facility, we normally have a lot of customized projects. So a lot of the functions do need to be developed in-house. And our bioinformatics team have been working really hard over the past 3 months to develop an in-house tool called MGI Kit. And with this MGI Kit, I think on the instrument tool from MGI, you can do single index support, dual index, but you can't really do much with UMI or mixed library types on the same line, or handle indexes shorter than cycles, do a bit of customization. And but with the tool developed by our bioinformatics team, you can achieve all of them. The speed is quicker. I'm not a bioinformatician, but it seems like 40 is bigger than 17. memory usage, it uses slightly like, it's not slightly, it's significant like a smaller memory, but we can't do parallelization here, which means you can only apply this MGI to a single core on your HPC, while with MGI2, I believe you can apply to multiple cores. And apparently, if you are comparing the libraries using Illumina FastQ headers, you need a bit customization, and we are happy to discuss more and have more conversation either with MGI and with other users about the data analysis and data formatting. So I think it's roughly about time. So this is our staff. As you can tell, we have staff. This is our genomics team. On the left-hand side, this is our bioinformatics team led by Dr. Jim Brain on the right-hand side. And I'd like to again thank our funding partners and the staff of SAGC. So as I mentioned, it's actually very straightforward to get in touch with us if you are interested, want to do some genomics and bioinformatics projects with us. Either just drop an email to our email address, sagc@samurai.com, visit our website, leave a message there. And if you are based at Adelaide, welcome to pop in on Samurai. We are located on North Terrace, level 5. And we also have a Flinders node and the White Campus node. You can directly reach out to our staff over there. And I think that's all my talk. And thank you very much. I will take questions at the end of the session.
Thank you, Sen. That was wonderful. Thank you very much for the feedback and also introducing South Australian Genomics Centre. So I hand it to Haofei to, I don't know, Haofei, did you want to have any comments around indexing in MGI or before you start your own presentation?
Yeah, I'll briefly introduce the Mgi sequencing platform like technology very briefly, and then respond to what Sen's experience with the Mgi instruments.
Okay. Great. So maybe I introduce Haofei first. Haofei received her PhD from the University of Queensland in bioengineering and biomedical engineering. After extensive experience in next-generation sequencing in her PhD studies, she joined MGI in 2020, and she improved her further hands-on experience and know-how of Mgi Sequencing Platform and whole genome sequencing, whole exome sequencing, RNA-seq related Ngs Applications. She's currently working as field application scientist to provide scientific advice and technical support for customers.
Thank you, Azadeh, for the introduction and for the introduction of the Mgi sequencing platforms as well. And thanks, Shane, for sharing his experience with the G400 sequencer. So I'm Haofei from MGI, and today I'd like to take the opportunity to introduce MGI's CoolMPS sequencing techniques. Before I start, I want to emphasize that this presentation is initial presentation of some internal testing result and some results from the collaborated projects. It's not the official released product information. So as Azadeh mentioned, the DNBSEQ G400 technology. Let me just adjust my desktop here. Yeah, so the DNBSEQ sequencing technology have a few unique features. So one is the use of DNB as the sequencing template. As the DNA nanoball is generated using linear amplification, which with every copy is synthesized using the original library as the template in a way that we greatly reduce the cumulative error. And the other feature is the use of pattern array flow cells, which allows a high density of the DNB to be incorporated on the surface of flow cells while minimizing the signal crosstalk between neighbor DNBs. So the combination of DNB-based template and plus the pattern flow cell, plus the CPAS sequencing technology, we're able to increase the sequencing accuracy, mainly the SMP and indel precision and sensitivity up to 99%, and then greatly reduce the duplication rate down to 2%. Here, I want to mention that the duplication rates, the better performance of duplication rate on the MGI platform is that the Illumina platform tend to have these optical duplicates issue. That's when the signal from the same cluster got wrongly assigned as two different reads that creates a duplicate from that's actually one clusters. And for the Mgi G400 sequencer, because the DNBs are assigned to a fixed position on the pattern flow cells, we minimize the crosstalk between the neighbor DNBs. So there's no such issue as much as what we observed on the Illumina platforms. And with this technology, we are able to greatly reduce the index hopping down to 0.0001%. So in addition to the CPAS technology, which we also term as the standard MPS, MGI also developed a CoolMPS sequencing techniques to further improve the sequencing accuracy. So the CoolMPS technology is a unique antibody-based chemistry which avoids the DNA scars that can accumulate with traditional sequencing methods. So instead of incorporating the fluorescent labeled nucleotides during sequencing, the CoolMPS uses nucleotides with natural bases for the second-strand synthesis, and then introduce fluorescently labeled antibodies for the base recognitions. So these four types of antibodies are base specific, so they will selectively bind to corresponding gene TP. And after imaging, the antibodies can be easily removed by introducing buffers having different pH, which leaves a natural basis for the next cycle of synthesis. So there's no scar left on the incorporated nucleotides. So let's break down to what happens at each cycle of the sequencing. So each cycle starts from base incorporations, during which the cold nucleotides with a 3-end extension blocks are introduced into the flow cells and then incorporated onto the template. And after that, we have 4 types of fluorescently labeled antibodies to be introduced and then selectively bind to the corresponding DNTP. And then before imaging, the excessive antibodies are removed and the flow cell is imaged for base calling. And at the end of the cycle is a regeneration steps, during which the antibodies and the 3-end extension blocks is removed from the templates and leaving a completely natural basis without any modification that's ready for the next cycle of nucleotide additions. So these cycles will be repeated for desired read lengths. As we know that the scars on the incorporated bases may suppress the fluorescent signals and limit the cycle completion performance, the use of natural bases would enable a higher enzyme fidelity and less systematic sequence-based errors. And since the sequencing reaction doesn't cause a scar remain on the DNA, it helps to reduce the scar-induced quenching in the CoolMPS sequencing. So from the figures on the right-hand side, we can see the signal intensity when the sequencer is reading through A, followed by C and G and T. So we can see that the signal from the 4 bases are at a similar level, and they are well maintained regardless of the identity of the previous bases. So this indicates that for one, the antibody can specifically bind to the corresponding nucleotides, and they can also be efficiently washed off before the next cycle of reading. And also there's low signal loss when switching between the 4 bases, which helps to keep a good signal when the sequencer is doing reads. the So the use of antibody offer another advantages. That is, more than one fluorescent labels can be conjugated onto one antibodies, which helps to provide a stronger signal and achieve a higher signal to noise ratio. This helps to achieve a more accurate base calling, and also consequently, it can potentially reach longer rates. So here we can see the comparison of the signal intensity between the standard MPS and CoolMPS. So the CoolMPS can reach almost doubles the signal from the standard MPS. So the cycle completion or reaction rate is another indication of the sequencing performance. So in traditional sequencing, when the synthesis goes on and on for many cycles, the DNA scar would increasingly affect the efficiency of chemical reaction, causing the base incorporation of some template molecules to either fall behind or goes one base faster. This is reflected as run-on and lag on the MGI platforms. So the run-on and lag would increase with more cycles of synthesis, causing the signal and sequencing accuracy to drop at longer read lengths. So from the result, we can see that compared to the standard MPS using labeled DNTP, incorporating the code DNTP onto the templates can greatly reduce the run-on and lag down to 0.1 and 0.2%. So with the CoolMPS, we can reach close to 100 reactions, which means less signal decays. So compared to the traditional sequencing method, the CoolMPS shows a better potential to achieve a longer rates. So here I want to present some of the internal testing on the CoolMPS sequencing platforms. This is the plot showing the changes of mismatch rate along with the increase of the cycles. Here the samples is the standard WGS library prepared using the human reference gene in NA12878. We can see the lines colored in orange comes from the Mgi Seq 2000 CoolMPS platforms. So the Mgi Seq 2000 is a previous name of the DNBSEQ G400 sequencers. And the dark lines are coming from the third-party platforms. So we can see up to 199 cycles. I think that's the end of a pair and 100 rates. The CoolMPS shows a much lower mismatch rate compared to the other platforms, which is corresponding to a higher accuracies. So this is the result of the sequencing performance across 10 different CoolMPS sequencing. That's from internal testing as well. So the figure at the top shows the total rates and Q30, and the figure at the bottoms are mapping rates and average error rates. So here the library we're sequencing is the MGI standard library, which is prepared using E. coli samples. So we can see that in average, the CoolMPS can reach a total rates of 480 million rates per lane, and Q30 is in average of 94%, split rate at 99.2%, and mapping rate up to 99.7%. And the average error rate is as low as 0.34%. So the standard variation is also very low, which suggests the performance of CoolMPS is quite stable. So this is the result comparing the standard MPS and CoolMPS across 7 different applications, including the general WGS, WGS PCR-free libraries, whole exome sequencing, RNA sequencing, and panel studies, and NIPT, and then CNV. So comparing the results from the 7 different applications, the CoolMPS shows an overall better performance compared to the standard MPS, especially for the whole exome sequencing and panel studies and also NIPTs. Next, I want to show some study cases coming from some collaborative projects. So the first one is a whole exome sequencing study case comparing the standard MPS platform and the CoolMPS platforms. So here the library prep is, the library are prepared using IDT exome V2 capture probes. In this study, we mainly observed the difference of the GC coverage comparing between the CoolMPS and standard MPS. So from the result, we can see that at the higher GC contents from 75% up to, I would say, 85%, the CoolMPS shows a better coverage compared to the standard MPS, as the normalized coverage is more closer to 1. And also for this study, we are comparing the ratio of soft clip rates on the third-party platforms and also the MGI sequencing platforms. So we can see from the result that the MGI platform shows a lower ratio of soft clip rates compared to the other sequences. So the ratio of soft clip rates shows the gap of unknown sequencing. So we can see that the Mgi sequencer can cover more regions compared to the other platforms. So the next study is a panel study comparing the CoolMPS to another platforms. So this study is mainly to look at the detection of variants having a very low mutation frequency down to 1%. From the list, we can see that for some of the variants that's having a very low frequency ratio, that's the other platform needs to detect that. The CoolMPS is able to detect those low frequency variants, and some of the results are quite close to what we expected. So yeah, this is another study case that's showing the whole exome study comparing the CoolMPS platform to the third-party sequencing platform as well. So the library is prepared using IDT capture probes, and we are comparing the sequencing with the depths of up to 700x. So when looking at the figures on the left-hand side, we can see that the parameters, including the Q30, mapping rates, and coverage are quite comparable between the MGI sequencing platform and the other sequencing platforms. But we see a lower duplication rate on the MGI sequencing platform that's down to 23%, while the other platform is showing up to 71%. Again, this duplication rate comes from a very high sequencing depths up to 700x. So I think this the difference in the duplication rate is corresponding to what Sen is experienced between the Illumina and MGI templates. And the reasons, as I mentioned, the Illumina platform tend to have a higher duplication rates comes from the optical duplicates. And similarly, when looking at the detection of low frequency variants, we can see that when the other platform fails to detect some of the variants, the MGI CoolMPS platforms can give some of the rating of those low frequency variants. So from the internal study and also some of the collaborative results, we can see that because the CoolMPS platforms gives more increased accuracy in the sequencing, it's quite useful for some of the applications when detecting a very low low mutation frequency of the variants. So it can be very useful for applications such as the whole exome sequencing and some of the panel studies and disease diagnostics. So for now, our G400 sequencer can support 2 types of flow cells for standard MPS. That is the small flow cells, which is in short is FCS, and also the FCL, which is the large flow cells. So the difference between 2 types of flow cells is that the small flow cell have only 2 lanes on the flow that's available for sequencing, and the large flow cell can support full length. So the large flow cell will generate more data up to 1800 millirates per flow cells. And for the small flow cells, we support array lengths from SE100, parent 100, and parent 150. And the large flow cells have more option in the read lengths from single and 50 up to single and 400, and parent 100 up to parent 200. As for the CoolMPS sequencing techniques, for now we're only supporting the large flow cells, and the rate lengths can be chosen from the single and 50, single and 100, and parent 100. And we're looking forward to the launch of CoolMPS sequencing kits supporting the parent 150 rate lengths. So the official product launch will likely be the end of this month or early next month. And MGI is offering a few of the trial opportunities so that we'll sequence your samples on the CoolMPS platforms for free. So if any of you are interested in this opportunity, feel free to either contact us or contact Sen for more details on the trials projects. And I'd also like to respond to Sen's question about the barcode and low split rate. So MGI platform does have a relatively lower tolerance on the low diversity regions. So we recommend to have a base distribution at least between 12% up to 65% per cycle. That is to have at least 10% or at least 10% of each basis in each cycles. So I think this issue is more not really comes from the imaging problem, but it's more related to the algorithm, and it's the base calling software that's embedded in the G400s. So this is a problem that we're constantly working to improve, and we've already managed to address this issue on the G50 sequencer, that's the desktop sequencer. But because the G400 is using a full channel imaging system, it got a more complicated imaging system and also this analysis systems. So we're taking more times to reach there. But in addition to that issue, if we can have the libraries to have a relatively balanced barcodes, we can reach the sequencing performance or even better sequencing performance compared to Illumina. So yes, that's in response to what Sen's been mentioned about the relatively low demultiplexing rates when using unbalanced barcodes. So that's all for the presentation from me today. Thank you for your time.
Thank you, Haofei. So just to follow up, so does it help if you spike in a balance library with
Yes,
if you have
spiking balance library would help as well, because the balance library will already have a balanced barcode that will help to save the low diversity region. As in Sen's previous slides, for some of the barcodes that only have 2 bases and have no, for example, it only have contains a basis of T and G and no A and T. And in that case, when reading through the 4 types of bases, we got no signal from A and T and a lot of signal from C and G. And that would be a bit difficult for the sequencer to distinguish the signals.
Okay,
Yeah.
thank you.
Thank you.
So we have a few questions from Rick in the Q&A box, which I'm going to ask you. So the first question is, what is the relative cost of the old approach and the antibody approach? So that I can answer that, it's the same. So that's, and the second question, can you recycle the antibodies? I don't think so, but
I don't think so, because
yeah,
the antibody will be used up during the sequencing. So at each cycle, we'll introduce new antibody reagents, and that at the end of that cycle, those reagents will be removed and washed off.
Okay, great. And then, so Rick has also asked, why are the CoolMPS cycles slightly longer?
Yeah, because the CoolMPS is using antibody-based chemistry, so it avoids the issues leaving a DNA scar at the end of each cycle. So the DNA scar will cause the chemical reaction to greatly lose its efficiency. That means at longer rates in the traditional method, at longer rate, maybe we'll have down to like 80% of the molecules that's doing the synthesis, so that significantly drop the sequencing accuracies. With the QMPS, we don't have the issue with the DNA scar anymore, so we can maintain the Q30 at a relatively higher rate. So in my previous slide, we can see the average Q30 is 94%. That means at longer rates, we can still maintain the sequencing accuracy to be at a relatively high level, so that allow us to read more cycles.
Thank you, Haofei.
Yes.
So that's all the questions in the Q&A box. We're
just one sec.
Yes.
I've got a question for Haofei. First of all, thanks Haofei for expanding like the indexing issue and the possible potential solution, which is to spike in more diverse indexed samples. I'm wondering, like, because the raw data file of the MGI instrument is a CAL file, right? CAL file. And from the CAL to FastQ, the algorithm is actually on the instrument by MGI, and I believe it's not an open sourced algorithm, right? And is it like possible for the team from MGI to work on the raw CAL to BCL like base calling algorithm to potentially increase the performance of demultiplexing?
I think so. We have a few like troubleshooting cases when like similarly customer would use only one barcode per lane, and that caused the issue with demultiplexing. So our bioinformatics team can reverse the demultiplexing using the CAL file only. So if you have such inquiry, I can talk to our bioinformatics, see if they can share the information with you, or either share a pipeline that you can work on the CAL file directly.
That would be great. Like actually our bioinformatics team are quite keen to work on like how exactly the base calling process is, and normally there is like room to improve the performance from the base calling process. So if you can put me in touch with the MGI bioinformatics team, we are happy to have a look to see whether we can potentially increase the performance.
Yes, of course. Yeah.
Okay, thanks.
Thank you. So thank you everyone for attending this webinar, and a special thanks to our speakers. Thank you, I really enjoyed that. So MGI marketing team will be in touch, sending all the registrants a recording of the webinar, and also some contact information in case you have any questions or you would like to trial the CoolMPS kit with South Australian Genome Centre. If there are no more questions, I'd like to thank everyone, and wish you a wonderful rest of the day. Thank you very much for attending.
Thank you.
Thanks. Bye.
Thank you. Bye.
Thank you very much.